Chapter 20, Integrative Physiology 2, Fluid and Electrolyte Balance. In this chapter, we'll talk about fluids and electrolytes and how they're important, or how it's important to maintain homeostasis of them. We'll discuss water balance, sodium balance, extracellular fluid volume. We'll also look at the importance of potassium and then how our behavior can change salt and water within our body, and how we control blood volume as well as osmolarity, and then we'll end with how we balance acid bases. So fluid and electrolyte homeostasis, why is that important? Well, our body is made up of water and electrolytes, so it's important to understand how they are regulated and why changes in their levels can adversely affect our body. So sodium and water. Extracellular fluid volume is made up of water and the solutes that are dissolved in them, especially sodium and other solutes as well, really impact osmolarity. And obviously there's that relationship between water and sodium. Water follows solutes, and the solute it follows the most is sodium. Well, what does potassium do in our body? We know potassium is an important ion. It's involved in cardiac and muscle function by helping to regulate our membrane potential. So we know our resting membrane potential is typically negative 70 millivolts. When we change potassium levels, we really impact excitability by causing the membrane potential to either depolarize closer to threshold or hyperpolarize further away from threshold. What about calcium? Well, calcium is important in many ways. It has a role in exocytosis, so it's an intracellular signaling molecule. It's involved in all muscle contractions, and it has other functions as well. For example, it's important in blood clotting. And then hydrogen ions and bicarbonate ions are obviously very important in our body's pH. So our body has to maintain mass balance, and we have different excretion routes, including our kidneys and our lungs, to be able to do that. So fluid and electrolyte homeostasis requires the integration of multiple systems. And so this is why it's an integrative physiology chapter, because we're looking at several organ systems working together. So the respiratory system, cardiovascular system, the renal system, as well as our behavior really influence fluid and electrolytes. So let's take a look at what would happen when our blood volume reduces. So when we have a reduction in blood volume, obviously that's going to reduce our blood pressure. Our baroreceptors, which sense changes in pressure, located in our aortic and carotid bodies, will sense that and send that information into the CNS. We will then trigger a homeostatic response to try to restore homeostasis. And so our cardiovascular system will try to raise blood pressure by doing what it does, activating the sympathetic output. And that will increase heart rate, increase contractility, which increases cardiac output. And it'll also stimulate vasoconstriction, which stimulates or increases blood pressure, or both of these increase blood pressure. Our behavior can also be altered. So our hypothalamus will start to make us feel thirsty, and that will allow us or cause us to seek out fluids. And the more water we bring in, the greater our extracellular fluid volume, and as a result, our blood pressure rises. And then our kidneys will be stimulated to produce produce less urine, to try to conserve the water that it has. And by doing that, we're not going to lower blood volume even more. So at least in that way, we'll be able to at least conserve what we have, and that will prevent a further drop in blood volume. Now, on the other hand, if blood volume is elevated, that's going to raise blood pressure. Again, our baroreceptors will sense that, and in this case, our cardiovascular system will stimulate the parasympathetic output. So when we stimulate the parasympathetic output, the sympathetic nervous system is not turned on. As a result, heart rate, parasympathetic stimulates the reduction in heart rate, but by turning off the sympathetic, we will not contract with more force, so our contractility will go down. We will not vasoconstrict. As a result, we will vasodilate. And we discussed this when we talked about the cardiovascular system. And all of that will help lower blood pressure. The kidneys will then be told to produce more urine, so we'll excrete our salts and our water, because water follows solutes, especially sodium, and that's where the salts come in. And by producing more urine, we will reduce our extracellular fluid volume, and that will help lower our blood pressure. And although behavior isn't written on here, we can, our behavior can change as well. We will not seek out more fluids in this case. Sometimes you just don't feel thirsty. So water balance in the body, we have to take in the same amount as we lose, and that will, that's the whole idea of mass balance. So we bring in water through the foods that we eat and the foods or the fluids that we drink. And don't forget, we also produce water through metabolism. So remember, at the end of the electron transport chain, that low energy electron, oxygen binds to it, and with hydrogen ions creates water. So we actually produce water, and we have different ways to lose water. So through our skin and our lungs, feces, but really primarily through urine. So this figure is really just to illustrate the fact that our kidneys can lose water. So we can lose water through the renal system, but our kidneys cannot produce more fluids. It can only conserve fluids. So it can't replenish fluids that we have lost. It can just conserve what we have. So what you're seeing here is fluid is going into the kidneys. So this would be like your nephrons. And so we can lose fluids by producing urine, but the best that we can do in terms of maintaining blood volume is to keep what we filtered. So we can just conserve. We actually can't replenish here in the kidneys. So let's take a look at the osmolarity of the filtrate that is observed inside the nephron. So here's a part of our nephron, so we're just not seeing the Bowman's capsule, but we know in the renal corpuscle, this is where the majority of filtration takes place, and the fluid that we filter has an osmolarity of 300 milliosmoles. In the proximal tubule, just to remind you, this is where we reabsorb and secrete. At the end of the proximal tubule, we still have an osmolarity of 300 milliosmoles. Now, as we go down the loop of Henle, and remember the entire loop of Henle we reabsorb, but we actually reabsorb different things in different parts of the loop of Henle. So in the descending limb of the loop of Henle, we will reabsorb only water. So now if you think about that, if our filtrate in the lumen of the nephron is losing water because it's getting reabsorbed back into our blood, we are only losing water. We are not changing the solutes. So if you just take water away, the osmolarity inside the filtrate or inside the nephron will start to rise. We're going to become more and more concentrated. And as a result, you can see the urine itself looks like it's getting darker and darker. And at the bottom of the loop of Henle, our osmolarity will rise to 1200 milliosmoles. As we go up the ascending limb of the loop of Henle, we will reabsorb ions. So no water is reabsorbed here. Instead, it's just ions, specifically sodium, potassium, and chloride. So if you are just losing ions now and water is not moving, you're only taking the solutes away. So now your osmolarity will start to drop. You're going to become less and less concentrated. So at the end of the ascending limb of the loop of Henle, so when you've completely ascended, your osmolarity will be as low as 100 milliosmoles. Now in the distal tubule, we will reabsorb and secrete. And then in the collecting duct, we will reabsorb and secrete. Now, what happens here in the collecting duct will really determine what the osmolarity of our urine will be. And it really is related to what we do with water. So our osmolarity of our urine that we excrete from the body will range from as low as 50 milliosmoles to as high as 1200 milliosmoles. So the 50 milliosmol urine would look clear, like water. The 1200 milliosmol urine will be very dark. So this is when your urine is really, really dark yellow. Okay, so I said the osmolarity of your urine is really determined by what happens in the collecting duct. So let's take a closer look here. The hormone vasopressin is going to play an important role. So vasopressin we've talked about before. It's also known as ADH or antidiuretic hormone. It's released by the posterior pituitary, and it acts here in the collecting duct. So when vasopressin is released, it's going to cause, it's an antidiuretic, so it causes you to not produce urine. So essentially what it does in the kidneys is it causes you to retain water. So if you retain water, you're going to increase blood volume. So let's see how it works. So when vasopressin is released, what it does is it causes water pores or aquaporins, or we can call them AQP2, to be inserted into the membrane of your collecting duct. And these are water pores, so obviously the name tells you what it does. It allows water to go through those pores. And by having these water pores, now we have a way for water to get reabsorbed. So we are going to reabsorb water. So as we bring the water out of the nephron into the blood, the filtrate in the nephron will, the osmolarity of it will rise and rise and rise. It's going to become more and more concentrated. And so it can get as high as 1200 milliosmoles. If we don't release vasopressin, then we don't have these water pores. So we will not reabsorb any water, so we'll just excrete what we had from the distal tubule. So our osmolarity can be as low as 50. Now here it shows 100, but it can go as low as 50 milliosmoles. So vasopressin, again, is released by the posterior pituitary, and here it's circulating in the blood. It will bind to vasopressin receptors here in the collecting duct. The vasopressin receptor is on the cell surface, and so when our hormone binds to a receptor on the cell surface, it's going to cause a very quick response. And typically what we do is we see second messengers start to appear. So it's basically a bunch of things get turned on inside the cell, and collectively we call them second messengers. And what they're going to do is they're going to tell these vesicles that have the water pores in them already, the aquaporin 2s, it's going to cause them to exocytose. And so when these vesicles exocytose, they're essentially going to insert the water pores into the membrane, and now water has a way to get reabsorbed into the blood. All right, so let's think about when we would want to release vasopressin. Now, the easiest thing is to think about, well, what does vasopressin do? So we said vasopressin causes you to retain water in the kidneys. So if you retain water in the kidneys, what will that do to your blood volume? So the more water you keep in the blood, your blood volume is going to go up. So when would you want to release vasopressin? When you want your blood volume to go up because your blood volume was too low. So when you have low blood volume, so here we see decreased arterial stretch due to low blood volume, then our hypothalamus will be stimulated to release vasopressin. Because when we have low blood volume and we release vasopressin through the posterior pituitary, we are going to reabsorb water back into the blood. And by reabsorbing water back into the blood, our blood volume will go up. So again, think about what vasopressin does. It causes us to reabsorb water. And so when we reabsorb water, what does that do to your blood pressure? Well, the more water we keep in the blood, the higher your blood pressure. So then when would you want to release vasopressin? You would want to release it when your blood pressure is too low so that you can raise it. So when you have low blood pressure, that will stimulate our baroreceptors to then tell our hypothalamus to release vasopressin. Now it says hypothalamus because the hypothalamus releases it actually through the posterior pituitary. So it will actually come out through the posterior pituitary, but again, you're going to conserve water, and that's going to increase blood volume, which we know increases blood pressure. Again, if you release vasopressin, you retain water. And the more water you retain, just water, then what would that do to your blood osmolarity? You would lower blood osmolarity. And so when do you want to release vasopressin to lower blood osmolarity? When your blood osmolarity is too high. Well, how high? When it's greater than 280 milliosmoles. So when your osmolarity, when your osmoreceptors in the hypothalamus sense that your blood osmolarity is higher than 280, it will also stimulate the release of vasopressin, which causes you to retain water, and that will help lower your osmolarity. So again, if you really understand what vasopressin does, and by keeping water, what that effect has on your body to your osmolarity, blood volume, and blood pressure, then it's easy to see when you would want to release it. So what the stimuli for its release would be. Okay, out of the three stimuli, blood volume, blood pressure, and osmolarity, blood osmolarity happens to be the most important. So what you can see is as osmolarity rises above 280, the higher it goes, you will release more and more vasopressin. It's a very strong stimulus, and that's because when we change blood osmolarity, remember, water follows solutes. And so when osmolarity changes, we're really going to start to change where water goes in our cells, whether it goes into our cells or out of our cells. And that's going to cause our cells to either shrink or swell. And we've already discussed how we really don't want that to happen to our cells because then they become dysfunctional. Okay, this figure shows what happens in the loop of Henle. It's the counter-current exchange. Don't worry too much about this. I wanted to keep it in just because it's, it's part of renal function. But this is just showing you how blood flow and the flow of your filtrate has to happen in the opposite direction. So in the descending limb of the loop of Henle, as the filtrate moves down, the blood supply is actually flowing up. And what that allows is it allows the reabsorption to happen all throughout this kind of length. If blood and the filtrate were moving in the same direction, so if you can see filtrate is going down, if blood was also flowing down, as soon as you reach equilibrium, you'll stop any kind of reabsorption. And so you won't be able to reabsorb all the way down the nephron. And the same thing happens on the other side. Okay. All right. This is just showing you what's going on in the ascending limb. So remember, in the ascending limb, we only reabsorb solutes, and the solutes we reabsorb is sodium, potassium, and chloride. And it's actually a symporter called NKCC. So N for sodium, K for potassium, and CC because it's two chlorides. This just looks at sodium balance, so the homeostatic response to salt ingestion. So if you eat a lot of sodium, a lot of salt, so you're eating saltine crackers, let's say, then you are increasing your osmolarity. Now, I said you're just eating saltines, right? Saltine crackers with a lot of sodium, but you're not drinking water. So we're going to say there's no change in blood volume. All you did was you brought in sodium, so osmolarity went up. And remember, an increase in osmolarity, especially when it's above 280, is a very strong stimulus for vasopressin release. So then vasopressin will be released. You will conserve more water in the kidney, so you'll reabsorb the water, and so you'll produce less urine. And that high osmolarity will also stimulate thirst. So the hypothalamus not only causes vasopressin release through the posterior pituitary, it also stimulates thirst. And you'll bring in more water, which will increase your extracellular fluid volume, increase blood pressure, returning kind of everything back to homeostasis. Speaking of sodium, let's talk about another hormone. So another really important hormone is aldosterone. Now, aldosterone is released by the adrenal gland, and the adrenals sit right on top of the kidneys. You have two of those. And what aldosterone does is it turns on your sodium-potassium pump. So there's our sodium-potassium pump. And when it turns on the sodium-potassium pump, it causes the kidneys to reabsorb sodium. So it causes you to retain sodium. And who follows sodium? Water. So as a result, you retain water. Now, aldosterone released by the adrenal gland acts here in the distal tubule. And so aldosterone is a steroid hormone, so it can cross the lipid bilayer because it likes lipids. So it will bind to receptors inside the cytosol. And remember, we talked about how if you bind to receptors inside the cytosol or inside the nucleus, you will turn on genes. So that's what's happening here. We are telling our DNA to transcribe into mRNA and then translate into new proteins. And so we'll make new channels and new sodium-potassium pumps and insert them into the membrane. And that will allow us to reabsorb sodium and secrete potassium. And when sodium gets reabsorbed into the blood, water will follow because water follows sodium. So that aldosterone is quite important, obviously, in regulating blood volume and blood pressure. And another hormone that also plays a role is the hormone renin. So renin is part of this system called the RAA. So it's part of the renin, angiotensin, aldosterone pathway, and it's really important in blood pressure regulation. So what renin does is it raises blood pressure. Okay, so we already talked about, this is our third hormone we've talked about in this chapter. The first one was vasopressin. The second one was aldosterone, and now it's renin. And notice all three of those hormones will raise your blood pressure. So they do similar things. So in this RAA, you'll see all of those hormones coming into play. So renin is released by your kidneys. And it's released by the kidneys when your blood pressure is low, because we already said renin raises blood pressure, so it would make sense that the stimuli to release renin is a drop in blood pressure. We'll also see, for example, the sympathetic nervous system can also stimulate the release of renin. When renin is released by the kidneys, it's released into the blood. It will come in contact with angiotensinogen. Now, angiotensinogen is constantly produced by the liver. So the liver produces angiotensinogen, and it's circulating in your blood, kind of minding its own business until the kidneys release renin. When renin is released, it will convert angiotensinogen into Ang1. Now, Ang1 is angiotensin 1. Angiotensin 1, once it's made in the blood from the angiotensinogen, will get converted into angiotensin 2. So the reason we would call something Ang1 is probably because there's an Ang2, and there is. Now, Ang1 will turn into Ang2 with the help of ACE. ACE stands for angiotensin converting enzyme because it converts angiotensin 1 to angiotensin 2. ACE is made by the endothelium of your blood vessels inside your lungs. Then Ang2 will do everything you can possibly think of to raise your blood pressure. For example, Ang2 is a vasoconstrictor because we know when we vasoconstrict our arterioles, blood pressure goes up. Ang2 stimulates the cardiovascular system to activate the sympathetic output, which we know increases the cardiac response. It increases heart rate, increases contractility, which increases stroke volume and cardiac output, and all of that raises blood pressure. Ang2 tells the hypothalamus to secrete vasopressin, which we know causes you to retain water in the kidneys, and by raising blood volume, you raise your blood pressure. Ang2 also tells the hypothalamus to make you feel thirsty, so we bring in more fluids, which we know raises blood pressure. And Ang2 tells the adrenal cortex to release aldosterone, which we just talked about, and that aldosterone causes you to reabsorb sodium. And who follows sodium? Water. So then you're going to retain water, which is going to raise your blood volume, and that will again contribute to high blood pressure. So basically everything that we've talked about that can raise blood pressure, this renin really starts it all. So this is why renin is such an important hormone, especially again in blood pressure regulation, this whole RAA pathway. All right, so what else can stimulate the release of renin? So again, a drop in blood pressure will directly stimulate the release of renin from those granular cells on the afferent arteriole, and we talked about that in the previous slide. We saw that. A decrease in your glomerular filtration rate can do that as well as the sympathetic nervous system through cardiovascular centers to increase renin release because, again, it raises your blood pressure. Now, what about a hormone to lower blood pressure? Now, this is called natriuretic peptides. And so when you have high blood volume, and as a result, it stretches your arteries in your blood. In your heart. Good Lord. So the myocardial cells, the heart muscles. So natriuretic peptide will do everything to help lower blood pressure, including telling the hypothalamus to release less vasopressin. And so when you release less vasopressin, you're going to retain less water. And so you'll basically lose more water, and that will help lower blood pressure. It'll tell the kidneys to decrease renin release. So renin helps to raise blood pressure, and so if you decrease renin release, it'll help lower blood pressure. It'll also tell the adrenal cortex to secrete less aldosterone, and that will also obviously occur because you're going to decrease renin release. And then it'll tell the medulla oblongata to decrease blood pressure, which we know is through the cardiovascular system. So it basically does the opposite of renin. So let's talk about potassium. So we already know potassium is important for cell excitability because of how it can change our resting membrane potential. And so we really want to regulate our potassium levels in a pretty narrow range. And aldosterone obviously is going to play a pretty important role because remember, aldosterone causes you to retain sodium, but when you retain sodium, you lose potassium because it does it by turning on the sodium potassium pump. So aldosterone is pretty important in potassium balance. Just to remind you, if you become hypokalemic, low potassium, it causes your resting membrane potential to hyperpolarize, meaning from -70, you'll become more negative. So you'll be further away from threshold, so your excitability decreases. So it can lead to things like muscle weakness and failure of your respiratory muscles in your heart. Hyperkalemia, which is an increase in potassium concentration, will cause your resting membrane potential to depolarize. You'll become closer to threshold, and so you become more excitable. So it can lead to things like cardiac arrhythmias. So causes of potassium imbalance includes kidney disease, diarrhea, and diuretics. So our behavior can also influence salt and water balance. For example, drinking a lot of fluids can increase blood volume, or not drinking will not increase fluid volume. So drinking can, be important in maintaining water balance. And also, what types of fluids are you drinking? Is it plain water, or does it have electrolytes in it? Our appetite also changes, for example, for salt. Sometimes we seek salty things, other times we don't. And then our behavior can also influence fluids. So we can become dehydrated when we don't drink and we go out and exercise in, the hot sun. For example, desert animals will avoid heat and kind of sit under a tree when it's really hot outside. So let's take a look at how our behavior and other things can influence blood volume and osmolarity. So in this figure, we're seeing osmolarity up here and volume over here. And so the middle box right here in the middle in green is a normal blood volume and a normal osmolarity. Now from this normal, let's say we decided to drink large amounts of water. If you are just drinking water, what is that going to do to your blood volume? It's going to increase it. So our blood volume, by drinking large amounts of water, our volume increases. But because you're drinking plain water, that's actually going to decrease our blood osmolarity. It's going to essentially dilute our blood because we're only bringing in water and no solutes. So hopefully this makes sense how to read this figure. Let's say you decide to eat a bunch of salty peanuts without drinking anything. Then our blood volume wouldn't change because right now we're not drinking anything. So this is kind of initially, what does this do? So just eating salty foods without drinking water will cause no volume change, but your blood osmolarity will increase. If you hemorrhage, if you are just losing whole blood, your blood volume will decrease. But your osmolarity won't change because you're losing whole blood. You're losing water as well as solutes. Okay, so hopefully this makes sense. Now this figure is a good summary figure. Obviously it's small here, so take a look at it in your textbook. But we've talked about how our cardiovascular system can influence blood pressure, how the RAA system can, how our kidneys play a role, and the hypothalamus. And this just puts all of those into one big figure. So nothing here is new, it's just we're seeing it all together. So, for example, when you become dehydrated and you have a drop in blood volume, it's going to lead to a drop in blood pressure. And that's going to cause a cardiovascular response that will raise blood pressure, primarily through activation of the sympathetic nervous system. The RAA will be turned on to try to raise our blood pressure back to normal. And our kidneys will play a role in fluid retention. And a hypothalamus, we know, already plays a role in vasopressin release and causing us to be thirsty. So, again, nothing is new here. We're just seeing everything working together. So, I would highly recommend you use this as a summary slide, as a test slide to see how well you connected and learned the material. And then let's end with acid-base balance. So normal pH of our plasma is about 7.35 to 7.45. Hydrogen ions obviously play an important role in pH because pH is power of hydrogen. And so our hydrogen ion concentration has to be closely regulated. And one of the reasons, there's many reasons why. One of them is the fact that when we change our pH, we can change or we can break bonds, chemical bonds. And so we know when we break bonds, we can change the structure of our proteins. And we started talking about this week 1 that our structure equals function. And so when we start to break the structure of our proteins, they don't work anymore. And so we're really going to disrupt body function. Abnormal pH affects the nervous system as well. Hydrogen ions actually block our sodium channels. And remember, an action potential is just the opening and closing of sodium and potassium channels. So the more hydrogen ions we have, the more sodium channels will be blocked. So our nervous system, our neurons won't be able to fire action potentials. So when we have acidosis, which is when we have too much hydrogen ions, our neurons as a result become less excitable. And so our central nervous system will become depressed. On the other hand, if we have alkalosis, we have less hydrogen ions than normal, then that means those sodium ions that are normally supposed to be blocked by the hydrogen ions are not. And as a result, we become hyperexcitable. pH disturbances are also associated with potassium disturbances because we have a potassium-hydrogen exchanger. So again, we see how important all of our ions are in our body. So what we really have to deal with are acids. That's why we keep talking about hydrogen ions. We really don't have to deal with too much bases. And the reason is because we make acids through metabolism. We also bring in acids through our diet. For example, fatty acids, amino acids. So the fats that we eat, the proteins that we eat, these are acids. Plus, we make acids through metabolism. We know that when we're doing anaerobic metabolism, we make keto acids. When you don't have enough glucose for your brain, then our liver will make keto acids. So we really don't want to disrupt our body's pH, and so we have to have ways to deal with all of these acids. So in the previous figure, I said normal pH is between 7.35 to 4.5, so that's what we're going to keep it at, okay? So if there's any questions related to pH, please stick with the 3.5 to 4.5 range. Now, there are 3 major ways we can deal with acids, and the first is buffer. So a buffer is basically anything that will bind a hydrogen ion so that that hydrogen ion doesn't change our body's pH. The most important buffer in our body is bicarbonate ion. Okay, so this HCO3 minus, most important. But again, anything that binds up hydrogen ions is a buffer. So certain proteins, hemoglobin, phosphates, these are all buffers in our body. Now, after having these buffers, we also have organ systems that play a role in trying to restore body pH. And the most important one is actually our respiratory system. So remember, CO2 in your body is like having acids in your body. So by changing our breathing, we can change our body's pH. If we want to raise our acid level, so we want to become more acidic, we want to decrease our pH, we'll conserve hydrogen ions. If we want to get rid of acids, we want to become more alkaline, then we'll breathe out more CO2. We'll increase our ventilation and expel more CO2. The other system is the renal system. So our kidneys will either retain or excrete hydrogen or bicarbonate ion as required. So if we are too acidic, it'll keep the bicarb and get rid of the hydrogen. If we're too alkaline, it'll keep the hydrogen and get rid of the bicarb. So again, to summarize, acids, that's really what we have to deal with because we bring it in through our diet and we also make them. Bases don't have to deal with as much. There are few dietary sources of bases. And how do we deal with changes in pH? Like we said in the previous figure, buffers are our initial go-to, the most important being bicarbonate ion. And then we have our respiratory system and then our renal system. So just to remind you of our respiratory response, when we become too acidic, our hydrogen ion concentration is too high, the chemoreceptors will cause our respiratory system to increase ventilation so that we breathe out CO2. If our CO2 level is too low, well, I don't see that here, but if it were too low, we would do the opposite. And then our kidneys. How do our kidneys respond? Our kidneys will either retain or excrete hydrogen or bicarb as required. And this will actually happen in the collecting duct. So in the collecting ducts, we have special cells called intercalated cells. And we have two types. We have a type A intercalated cell and a type B intercalated cell. And it's quite easy. The type A cells will respond if you have acidosis. So if you are too acidic. Type B will respond if you have alkalosis or you're too basic. So A for acid, B for basic. So, it looks quite complicated, but all you really need to know is that if you have too many acids, your pH is too low, then the type A cells will excrete the hydrogen ion and keep the bicarb. Exactly what you need it to do. If your body is too basic, you are too alkaline, you have too little acids, then your type B cells will do exactly what you need to do. It will get rid of the buffer, the bicarb, and it will keep the hydrogen ion. Okay, and just to go over this, changes in pH, acidosis and alkalosis can happen due to a metabolic reason or a respiratory reason. And so when we talk about someone's blood pH changing, we don't typically just say that they have acidosis or alkalosis. We specify whether it's respiratory acidosis or metabolic acidosis. So if you have respiratory acidosis or alkalosis, it's really due to a problem with your respiratory system. So remember, CO2 is like acids, right? So having more CO2 in your body is like having more acids in your body. And so when you have a problem with your respiratory system, you're going to alter CO2 levels, and that will alter your pH. And so respiratory acidosis occurs when you are keeping too much CO2 in your body. Respiratory alkalosis occurs when you are losing too much CO2 from your body. Now, metabolic acidosis and alkalosis is basically anything that's not CO2 related. So making too much lactic acid, making too much keto acids. So those reasons, if those are the reasons why you have changes in pH, then it's a metabolic issue. So if you have a change in pH because you have kidney failure and your kidneys are not working the way that they should, then you would have metabolic acidosis. And that is it for this chapter. Thank you.